خلقكم من نفس واحدة ثم جعل منها زوجها وأنزل لكم من الأنعام 8 أزواج يخلقكم في بطون أمهاتكم خلقا من بعد خلق في ظلمات 3 ذلكم الله ربكم له الملك لا إله إلا هو فأنا تصرفون.
First of all, what is recombinant DNA technology? Recombinant DNA technology or genetic engineering is a method to produce gene manipulation with desirable traits. Here is the important thing to remember is the tools used in the recombinant DNA technology. First is target DNA, restriction enzyme, vector, host cell, and modifying enzyme. Let's go through one by one, you all.
So, the first one is target DNA. Target DNA refers to the specific DNA segment that we want to isolate or maybe the gene of interest.
The next one is restriction enzyme. So, this enzyme is used to cut or cleave the DNA at restriction site in 2 ways, which is staggered and straight, that resulting a sticky end and a blunt end. This enzyme also known as endonuclease or molecular scissors.
Next is cloning vector. This vector we call as rumah singgah because they are able to carry a foreign DNA into a host cell but can't stick with them all time. Sad, huh? So, example of cloning vector that we use widely is bacterial plasmids. So the foreign DNA is inserted into a cloning vector which then replicate into a host cell.
Now back to host cell. What is host cell? It's a typically like a house for a foreign DNA and cloning vector to replicate. This host cell allow vector to multiply themselves. Example of host cell is bacteria E. coli, yeast cell, and mammalian cell. This host cell also can maintain structure of the recombinant DNA from one generation to another.
Finally, let's talk about modifying enzyme. This enzyme helps join DNA fragment by catalyzing the formation of broken phosphodiester bonds. For example, DNA ligase repress gaps in the DNA backbone, while PEG polymerase is used in PCR, a method to amplify DNA segment.
That's all. I hope you guys can remember all the tools used in recombinant DNA technology. Now, let's move on to the next topic of the process of gene cloning. Gene cloning is defined as a technique used to produce a group of individual cell or DNA with identical genetic makeup. This technology is definitely helpful for us in many aspects in life, such as for mass-producing insulin, growth hormone, which is why this technology is usually used by researchers in the biomedical field.
There are a few steps in accomplishing this process. First is to isolate the cloning vector, which is usually plasmid from E. coli bacteria, and the target gene. There are 2 known methods in isolating the gene of interest, which is genomic cloning and producing complementary DNA. Genomic cloning is the process of cutting genes from a complete chromosome using restriction enzymes into thousands of fragments. After that, to separate the gene of interest from all the thousands of fragments of genes that are cut, there are 2 methods, which is gel electrophoresis, a method for separation of various fragments of DNA based on their size and charge. There is also the method of using DNA probe to locate target gene and reveal it through the X-ray film.
Next, in the second stage, the process is to cut or cleave both the target gene and gene vector using identical restriction enzyme to provide a complementary sticky or blunt end between the gene of interest and plasmid.
Then, on the next stage, which is the third stage, gene of interest would be treated with the same restriction enzyme, EcoR1, as the gene vector. DNA ligase enzyme would then also catalyze the covalent bonds formation to join the 2 molecules into one new recombinant plasmid. Plasmids from the commonly used E. coli have 2 marker genes, which is resistance ampicillin and the LAC gene.
After that, the transformation will happen in stage 4. The purpose of this stage is to introduce recombinant plasmid into bacterial cell. Firstly, the mixture of plasmids is mixed with bacterial cells in a medium containing calcium chloride, followed by a short heat stop that helps to open pores in the plasma membrane to allow the plasmid to enter.
We are now at the final stage, stage 5 and stage 6. In this stage, bacteria with the recombinant plasmid are allowed to reproduce, cloning the inserted gene. Bacteria that contain the plasmid with an intact LAC gene will appear blue. In contrast, colonies with the recombinant plasmid will turn white because the LAC gene is non-functional.
Okay guys, we shall move forward to another interesting topic, which is polymerase chain reaction PCR. PCR is another way of making copies of a specific section of DNA without the need for vectors or whole cell. Basically, it is a method to amplify DNA in vitro. As you guys can see, these are the components required in PCR, which is the PCR machine itself, DNA fragment, DNA primer, free DNA nucleotides, tag polymerase, and also mix buffer.
Lastly, for this subtopic, we will see the stages in PCR. So, PCR requires only 3 stages. The first one is called denaturation, which separates the 2 nucleotide strands of the DNA molecule. This process happens at 94 to 96 degrees Celsius. The next stage is annealing, which will happen at 68 degrees Celsius. So, the DNA fragments were cooled down, the primers bound to the single-stranded DNA. Tag polymerase and primers were also added. The last stage is extension or elongation. This stage happens at 72 degrees Celsius. The temperature is increased to begin the synthesized new DNA strands. Free nucleotides are also added to the primer's 5' to 3' direction to form a double-stranded copy of the target DNA.
Let's move on to the application of recombinant DNA technology. We will now explore specific examples of where biotechnology is used in pharmaceutical, agriculture, and the environment. But before that, I will explain about cDNA. What is actually cDNA? cDNA, or complementary DNA, is a DNA that is made in vitro by using mRNA as a template and enzymes reverse transcriptase. cDNA has no introns and only contain exons. Do you remember what is introns and exon are? Intron is a non-coding segment, while exon is a coding segment. Actually, cDNA is used in the production of insulin by transgenic bacteria, which is E. coli, to treat diabetes. So, basically, insulin is produced by using cDNA instead of DNA. This is because DNA has introns, which is non-coding segments, and will need to undergo RNA splicing. So, how to synthesize cDNA? First, mRNA for insulin is isolated or extracted from beta cell islets of Langerhans in human pancreatic cell. Second, reverse transcriptase enzyme is added to synthesize the first cDNA strand by using mRNA as a template. Third, mRNA strand is then degraded by using mRNA degrading enzyme. After that, the first cDNA strand is used as a template to synthesize the second cDNA strand by using DNA polymerase enzyme. So, we will now have both strands forming a double strand, which is complementary for each other. Now, I will explain the steps in producing insulin by using cDNA that we have synthesized early. So, we will have to understand each task. What is each task? I stand for isolation, C is for cleave or cut, another I is for insertion, and T is for transformation, A is for amplification, and S is for screening. So, first, isolation. Plasmid is isolated from bacterial cell, which is E. coli, and is used as a cloning vector. Second, cleave or cut. Both ends of double-strand cDNA and plasmid are cut by using the same restriction enzymes, which is specifically PEM-high, to produce compatible sticky ends. In insertion, the gene of interest, which is insulin gene, is inserted into the plasmid, and they are joined by using DNA ligase. For transformation, the recombinant DNA is introduced into host cell, which is E. coli, through transformation process. In amplification, the recombinant DNA is amplified in the host cell to produce many copies of recombinant plasmid. And lastly, screening. Bacterial cell with non-recombinant plasmid will form blue colonies, while bacteria with recombinant plasmid will form white colonies. After screening, we will only culture white colonies for the production of insulin hormone because it has recombinant plasmid.
Genetic modification using recombinant DNA technology is revolutionizing the environment and agriculture. First, in agriculture, GM plants like glyphosate-resistant soybeans survive herbicides, while BT maize produces a protein that protects against pests. Second, transgenic animals like faster-growing salmon are engineered for better growth and disease resistance. At 18 months, genetically altered fish are 24 inches long and weigh 6.6 lbs, while normal fish are only 13 inches and 2.8 lbs.
GM bacteria, like Pseudomonas, are used to clean oil spills by breaking down hydrocarbons. GM mosquitoes are also being developed to control malaria and dengue.
Next, we will move on to the importance of insulin produced by genetic engineering. First, we can produce insulin in a large amount. And don't worry, it is compatible to human insulin, so will not have side effects. And most importantly, it is halal and cost efficient.
Biotechnology is a rapidly evolving field that impacts many aspects of our lives. Today, we'll take a closer look at both the advantages and disadvantages of biotechnology in agriculture and medicine. Firstly, bioengineered plants grow faster, are stronger, and need fewer chemicals like herbicides and fertilizers, making farming more sustainable.
Secondly, GM foods are more nutritious, last longer, and are cheaper to produce, which can help reduce food costs. Thirdly, biotechnology in medicine allow for the production of important medicines, such as vaccines and hormones, which can be life-saving, especially in poorer regions. While biotechnology has many benefits, there are also some downsides to consider.
First, GM crops can sometimes lead to overuse of chemicals, which might harm the environment and create resistance to pesticides. Another issue is the uncertainty about the long-term effects of GM foods on our health and the environment. Additionally, companies often patent GM seeds, which means farmers have to buy new seeds each year, increasing costs. Lastly, there is always a risk that genetic changes could have unexpected effects on